So back in the 1950s, doctors were testing a new drug on tuberculosis patients in a clinical ward.
Right, the lung treatment.
Yeah, exactly. The drug was, designed to clear up their lungs.
Yeah.
But almost immediately, the medical staff noticed this entirely unexpected side effect.
They weren't just breathing better.
No, they were euphoric. I mean, people who had been bedridden with a lethal respiratory disease were suddenly hyperactive, socializing, and practically dancing in the halls.
Which is just a bizarre historical accident.
It really is.
So,
But it actually gave us the modern blueprint for understanding depression. But today, the future of treating that depression might not come from a pharmaceutical lab at all.
no, it might come from a bizarre-looking fungus growing on the side of a dead tree.
Right. So you want the real hard science on how to optimize your brain health, but you want it translated into something you can actually use. That is our mission today on this deep dive.
Absolutely.
We are taking a dense, really fascinating 2020 scientific review published in the International Journal of Molecular Sciences titled Lion's Mane Mushroom, a therapeutic review for depressive disorder. And we are extracting the most critical, mind-blowing insights just for you.
It is a remarkable piece of scientific literature. The core question it explores is profound, really. Could this strange-looking fungus, which is also known as Yamabushitake, or the monkey's head mushroom, legitimately be a new frontier in treating depressive disorders?
Which is a huge claim.
It is. We are looking at a potential paradigm shift where we might, substitute the heavy, often difficult side effects of pharmaceutical drugs with natural neurobiology.
Okay, let's unpack this.
Yeah.
Because to understand why neurologists are looking at a mushroom, we have to look at the staggering reality of depression right now.
Right.
We are talking about a neuropsychiatric disorder that affects over 300 million people globally.
It's massive.
It crosses all demographics, all ages. It is one of the absolute leading causes of the global disease burden. And the traditional medications we rely on, they have a surprisingly high failure rate.
Yeah, and that failure rate is exactly why this research is being funded in the first place.
Yeah.
If you look at the clinical data on currently available antidepressants,
Like SSRI.
specifically SSRIs and SNRIs, yeah. In adults with moderate to severe depressive disorder, those medications only successfully relieve symptoms in about 20% of patients.
Wait, 20%.
Just 1 in 5 achieve full remission of their symptoms.
Oh, wow.
And furthermore, over 50% of antidepressant users report experiencing severe, life-altering side effects.
Like what?
We are talking about metabolic changes leading to weight gain, extreme dizziness, severe anxiety, and significant sexual dysfunction, which includes decreased interest and the physical inability to reach orgasm.
I imagine those side effects are the main reason people just abruptly stop taking their medication.
Precisely. You have a high failure rate combined with a really heavy burden of side effects. That equation is what drives the medical community to look toward alternative functional foods.
Like the lion's mane.
Right. Researchers are searching for novel therapeutic compounds in things like lion's mane, which has a very long history in traditional East Asian medicine, to see if we can achieve better efficacy with, better tolerability.
Oh, and so going back to that tuberculosis story from the 1950s, are you saying our entire modern understanding of brain chemistry was basically a side effect from a lung treatment?
In many ways, yes. The tuberculosis drug was called aproniazid. When scientists investigated why it was making patients so euphoric, they realized the drug was acting as an inhibitor.
How so?
Well, in your brain, you have an enzyme that basically acts like Pac-Man, going around and chewing up excess neurotransmitters. Aproniazid stopped that Pac-Man enzyme from working.
Oh, I see.
This meant it was flooding the patient's brains with monoamines, specifically serotonin, dopamine, and norepinephrine.
And researchers realized that if flooding the brain with these chemicals causes euphoria, then a lack of these chemicals must cause depression.
Exactly. And they confirmed it shortly after with another drug called Rezorpine, which was given to patients for hypertension.
And it did the opposite.
Right. Rezorpine did the exact opposite. It depleted the brain's stores of serotonin and dopamine, and it induced a severe syndrome that looked exactly like clinical depression.
Which became the foundation for the monoamine hypothesis.
Yes.
It's the idea that depression is caused by a physical deficiency in specific neurotransmitters. Let's visualize how this actually works. Think of the communication between your brain cells less like a physical wire and more like two people trying to send text messages to each other across a gap.
I like that analogy.
Right. The first brain cell sends a chemical text message like, hey, feel happy about this beautiful sunset. across a space called the synapse. Serotonin and dopamine are the actual text messages.
Right.
But in a depressed brain, the signal strength is like a dropped cell phone signal. It's too weak. Not enough messages are being sent, or they are being deleted before they cross the gap. The message just never arrives.
That is an accurate way to describe the synaptic cleft. But modern neuroscience makes it clear that depression isn't just a dropped signal.
That's more than that.
It's far more complex than just low serotonin. Which brings us to the second root cause of depression, which is the neurotrophic or neurogenic hypothesis.
Meaning brain growth.
Exactly.
Mhm.
This hypothesis suggests that depression is fundamentally the brain losing its physical plasticity. Under severe or chronic stress, the brain loses its ability to adapt and grow.
Oh, wow.
Specifically, we see a sharp decrease in a protein called brain-derived neurotrophic factor, or BDNF. BDNF is essentially a crucial growth fertilizer for your brain.
Okay, so without the fertilizer.
When BDNF levels drop, the hippocampus, which is the core area of the brain responsible for memory and emotion, can actually physically shrink in volume.
That is terrifying.
Right.
The brain literally loses its ability to maintain its own structural integrity.
Yes, it physically atrophies.
And then there's the third cause, which I think is the most surprising, honestly, the inflammatory hypothesis. Scientists are finding that depressive disorders are heavily linked to a massive increase in pro-inflammatory cytokines, right?
Yes, specifically immune system proteins like TNF-alpha and interleukin-6.
Right.
When we look at animal models, if researchers administer inflammatory agents like a molecule called lipopolysaccharide, or LPS,
What does that do?
it essentially tricks the body into thinking it has a massive bacterial infection, and you can reliably induce depressive-like behaviors in the animals within hours.
That's from inflammation.
Yes. They stop eating, they lose interest in sugar water, their motor activity drops to almost zero.
Wow.
And we see this mirrored in clinical trials where healthy human volunteers were injected with LPS. As their physical inflammation spiked, they reported an acute, severe increase in psychological depressive symptoms.
I want you, the listener, to really think about the last time you had a terrible case of the flu. Think about how that physical sickness in your body made you feel mentally.
It's a very recognizable feeling.
Yeah, you didn't just feel physically achy. You felt mentally exhausted, apathetic, and like completely drained of joy. You probably didn't want to talk to your friends or do any of your favorite hobbies.
Right.
That sickness behavior is an evolutionary response to keep you isolated so you don't spread infection, but it is also the inflammatory hypothesis in action. Your immune system is firing off those cytokines, and your brain translates that physical fire directly into mental depression.
Which perfectly sets the stage for our central question. We now have these three distinct biological failures causing depression.
The dropped cell signal at the synapse, the lack of physical brain fertilizer, and a raging inflammatory fire.
Exactly. How does one specific species of mushroom counteract all three?
Because if it only targets the serotonin, it's really just doing what current pharmaceutical antidepressants already do.
What's fascinating here is that lion's mane contains highly unique neurotrophic compounds, specifically classes of molecules called hericenones and erinacines.
Right.
The critical detail about these specific compounds is their molecular weight and structure. They are lipophilic and incredibly small, which allows them to actually pass directly through the blood-brain barrier.
And the blood-brain barrier is notoriously difficult to cross. I mean, think of it less like a solid wall and more like a microscopic, highly restrictive coffee filter wrapped around your brain's blood vessels.
That's a great way to picture it.
Most standard drug molecules are big, bulky macromolecules. They are too chunky to pass through the mesh of the filter, which is great for keeping out toxins, but terrible for getting medicine into the brain.
Right.
But these mushroom compounds, the erinacines, are so tiny they slide right through the coffee filter directly into the brain fluid.
And once they cross that barrier, they begin to address those three root causes. Let's examine the first mechanism, which is restoring the monoamine signal.
Okay, so researchers set up a study where they took mice and subjected them to chronic restraint stress for 14 straight days.
Which is intense.
Yeah, this essentially induced a deep clinical depressive state in the animals, which severely depleted their serotonin, norepinephrine, and dopamine levels.
Right.
But then, researchers administered an extract from the lion's mane that was highly enriched with a specific compound called erinacine A.
And the results are striking.
Yeah, it didn't just mask the symptoms, it actively modulated the chemistry in the hippocampus and successfully restored those depleted neurotransmitters back to baseline levels.
And beyond just restoring the chemical signal, it addresses the second cause, the neurogenic hypothesis, by acting as a structural fertilizer.
The brain growth part.
Yes. Lion's mane profoundly stimulates nerve growth factor, or NGF. In studies where animals were chronically given high doses of the mushroom extract, researchers observed a significant increase in actively proliferating cells in the hippocampus.
Wait, I need a bit of clarification there. How do researchers actually know new brain cells are being born? Do they just, like, count them under a microscope?
Well, they look for very specific molecular signatures like pCNA and Ki67.
Okay.
You can think of these markers as cellular birth certificates. These proteins are only expressed in the nucleus of a cell when it is actively dividing and multiplying.
I see.
When scientists observe a high presence of these birth certificates in the hippocampus of the mice-fed lion's mane, they have definitive proof that the brain is undergoing neurogenesis. It is literally glowing new neurons, and, crucially, those new neurons are surviving despite the stressful environment.
Okay, so we have the chemical signal restored and new brain cells growing. What about the third front, putting out the inflammatory fire?
The anti-inflammatory mechanisms were observed by isolating a different lion's mane compound called amicinone. Researchers took mice and gave them this amicinone extract about an hour before injecting them with that LPS agent we discussed earlier.
The agent that causes the massive cytokine spike, the fake bacterial infection.
Correct. The amicinone actively intercepted the immune response. It stopped the massive rise of the pro-inflammatory cytokine TNF-alpha.
Which is huge.
And perhaps even more importantly, it simultaneously boosted the levels of interleukin-10, which is an anti-inflammatory cytokine.
Oh, wow.
Yeah, it acts as both a shield against the initial inflammatory attack and a biological cooling agent to bring the immune system back to a resting state.
Okay, I have to jump in and push back on this a little bit.
Go ahead.
Wait, so it fixes the chemicals, it physically grows new brain cells, and it stops immune inflammation. That sounds like an absolute magic bullet. Like, it sounds like a supplement infomercial.
Yeah.
What does this actually look like when tested on living, breathing subjects and not just looking at cellular birth certificates in a lab?
That is the exact skepticism we should apply. It is why we have to carefully examine both the preclinical animal behavioral models and the human clinical trials. We have to see if this cellular activity actually translates into a measurable reduction of depression.
Let's look at the preclinical behavioral data first. In the animal models, scientists didn't just measure brain chemicals. They measured behavior using something called the forced swim test and the tail suspension test.
Right.
In these tests, an animal is placed in a mildly inescapable, stressful situation, and researchers measure immobility time. Immobility is essentially when the animal stops trying to escape.
It's a giving up behavior.
Exactly, which is a universally accepted marker of depression in these laboratory models.
And the results were statistically significant. The mice that were pre-treated with the lion's mane extracts showed drastically reduced immobility times compared to the control group.
They kept swimming.
They didn't give up. The compounds created a measurable behavioral resilience to stress.
Which paves the way for the clinical data in humans. And this is where the science gets really fascinating, though definitely more complicated.
Absolutely.
There was a notable study published in 2010 by Nagano and colleagues. They took 30 menopausal women and gave them cookies, literally baked cookies that contained 0.5 gram of lion's mane fruiting body powder.
Baked right in.
Yeah. The women ate these cookies every day for 4 weeks. At the end of the trial, they used a standardized psychological index, and the women reported significant alleviations in subjective symptoms of depression, generalized anxiety, frustration, and even physical palpitations.
We also have a more recent 2019 study by Vigna and colleagues. This one looked at 77 adults who were classified as overweight or obese.
Okay.
For 8 weeks, these volunteers were given capsules containing a mix of lion's mane mycelium and fruiting body extracts. The results from their psychological evaluations showed significant improvements in sleep disorders, baseline anxiety, and depressive symptoms.
Bullet, I can hear the hesitation in your voice.
This raises an important question about the rigorousness of these early human trials. As analysts, we have to look for the flaws in the data.
Right, we can't just take it at face value.
Exactly. The Nagano cookie study, for example, is highly intriguing, but an N of 30, just 30 people is a very small sample size.
Yeah.
Furthermore, it was entirely gender-specific by design, and the subjects were undergoing complex hormonal changes due to menopause.
Right. Menopause has its own massive impact on mood and brain chemistry. which makes it hard to say if the mushroom is treating clinical depression
Hmm.
or just smoothing out a hormonal fluctuation.
Exactly. The scientific review also highlights a case study of an 86-year-old man with recurrent depressive disorder who showed major cognitive and mood improvements after taking a lion's mane supplement for 6 months.
That sounds promising.
It does, however, that data is heavily confounded. Why? Because the patient was simultaneously taking mirtazapine, which is a powerful standard pharmaceutical antidepressant.
Oh,
Yeah, so we cannot definitively say if the mushroom, the pharmaceutical drug, or the synergistic combination of both caused the improvement.
there was also that pilot study done on female undergraduate students. They were given the extract for 4 weeks and reported improved anxiety and sleep quality. But the authors of the study specifically noted these students were right about to take a massive, high-stakes national examination.
Right.
So were they treating chronic neurological depression or just taking the edge off of acute exam stress?
That is the crucial distinction. Acute stress is very different from chronic major depressive disorder.
For sure.
Furthermore, with the Vinya study on overweight adults taking capsules, we have to heavily account for the placebo effect.
Because depression is notoriously susceptible to placebos, right?
Highly susceptible. Often, simply the act of receiving care, being enrolled in a study, and taking a capsule every day instills a sense of hope in a patient. And hope itself chemically alters the brain.
Wow.
The authors of the review stress heavily that while these early results are incredibly promising, we desperately need large-scale, double-blind, placebo-controlled trials across diverse demographics to truly prove clinical efficacy.
So acknowledging that we need better, much larger human trials, it makes me wonder what exactly the scientists should be testing moving forward. Because we aren't talking about a synthetic drug where a laboratory stamps out one identical molecule in a pill.
No, lion's mane is a living, incredibly complex organism.
Right. Should we be telling people to just eat the whole mushroom? Or should we be trying to isolate the most active compounds in a sterile lab?
That debate is the absolute cutting edge of the research right now. And it comes down to understanding the physical anatomy of the mushroom, because the anatomy dictates the chemistry.
Here's where it gets really interesting. When you look at lion's mane, you are basically looking at two distinct parts.
Correct.
You have the fruiting body, that's the part you can actually see growing on the bark of the tree, the part that looks like a white shaggy waterfall or a monkey's head.
The recognizable part.
Exactly. But then you have the mycelium. The mycelium is the vast root-like network hidden underneath the surface, digesting the wood. Researchers have found that these two parts contain entirely different arsenals of chemical weapons.
They do.
The hericenones, specifically hericenones A through K, are synthesized exclusively in the fruiting body, but the arenacenes, mostly A through I, are extracted almost entirely from the underground mycelium.
And both classes of compounds, the hericenones and the arenacenes, have been shown in vitro to successfully promote that vital nerve growth factor we discussed.
Okay.
But because they are partitioned into different anatomical parts of the organism, it leads to a massive debate in the scientific community regarding therapeutic synergy.
Synergy meaning how the chemicals interact with each other.
Yes. In a pharmaceutical laboratory setting, the standard operating procedure is reductionist. It is highly tempting to isolate the single most potent compound, say, arenacine A, from the mycelium, and concentrate it into an enriched, purified extract.
Like we saw with the stressed mice.
Exactly. As we saw with the stressed mice whose serotonin was restored, that targeted approach works beautifully. However, the review strongly suggests that the crude extract might actually be superior.
By crude extract, you mean literally just taking the whole natural mushroom fruiting body and mycelium together and extracting everything at once.
Correct. The data suggests that the unrefined, whole mushroom extract containing all its various known and unknown compounds might be just as effective, if not more so, than the purified isolates.
Why is that?
This is due to the synergistic effects, sometimes called the entourage effect. All those different molecules are essentially playing together like an orchestra, supporting and enhancing each other's absorption and efficacy, rather than just a single solo instrument trying to do all the work.
Have scientists definitively identified the single magic compound yet? Like, is there one specific molecule inside that orchestra that is acting as the conductor, doing all the heavy lifting?
The short answer is no, and the reality is they likely never will find just one single magic molecule. Yeah, because they are constantly discovering novel compounds within the fungus. The review highlights recently identified molecules with incredibly complex structures, We are talking about newly discovered sterols like ergosterol peroxide, cerevisterol, and another highly specific one called 3-beta, 5-alpha trihydroxyergosta 7022-denyne-61.
Okay, I'm not even going to try to pronounce that last one.
The nomenclature isn't the important part. What matters is that all of these newly discovered highly complex compounds also exhibit nerve growth factor-inducing activity.
So they all work.
Yes, when applied to cells, they all promote neurite outgrowth, which is the physical branching out of the brain cells. This proves just how breathtakingly complex this functional food really is.
Wow.
A single lion's mane mushroom is essentially a complete biological pharmacy unto itself, and we are only just beginning to catalog the inventory on the shelves.
So what does this all mean? If we zoom out and synthesize this entire stack of research for you, the listener, it comes down to this. Our current pharmaceutical approach to depression is flawed. SSRIs worked for some, but they fail many, often carrying punishing, life-altering side effects.
Right.
We now understand that depression is not just a simple chemical imbalance. It is a multi-headed biological beast involving monoamine deficits at the synapse, a physical loss of neuroplasticity and brain growth, and a chronic inflammatory fire in the immune system.
All three.
And sitting quietly in the forest is lion's mane, an ancient organism containing a highly unique blood-brain barrier-crossing cocktail of compounds. It's a cocktail that actively fights on all three of those biological fronts simultaneously.
Which is incredible.
The human clinical trials are definitely still in their infancy, and we need much more rigorous data to account for the placebo effect. But the underlying cellular mechanisms are undeniably real and intensely powerful.
If we connect this to the bigger picture, it leaves us with a truly profound philosophical question.
I'm intrigued.
It is a concept of evolutionary compatibility. Consider this. Fungi are an entirely different kingdom of life.
Right.
The fungal kingdom diverged from the animal kingdom over 1 billion years ago. And yet this specific fungus naturally produces highly complex molecules like erinacines and hericenones that perfectly, seamlessly bind to mammalian neurological receptors.
That's wild.
They fit our brain's biology like a key sliding into a lock, perfectly shaped to stimulate our brain growth and regulate our mood.
Wow.
It forces us to ask a deeply compelling question. Is this just some bizarre, random biological coincidence? Or is this evidence of a deeply ancient, forgotten symbiosis between the fungal kingdom and the mammalian nervous system?
That's a huge idea.
A mutual relationship that we, in our modern, hypermedicalized search to optimize our brain health, are only just now beginning to rediscover.
Now that is an incredible thought to leave on. Thank you so much for joining us on this deep dive. Keep questioning the world around you. Keep looking for the how and the why behind the facts, and keep learning. We'll see you next time.